Environmental is Contract Council and the CASM Focused Investment Partnership to do macroinvertebrate monitoring at a whole lot of different restoration sites for projects that are being done across a period of about 6 years. Thank you. And so some of this sort of thematically is probably going to be very old hat to some of you. For others, it'll be brand new and exciting, and you'll be revitalized, and it's going to change your life forever. Or at least hopefully you'll be interested because you all slide into a food coma from the carbohydrates one way or another. So without further ado, we answer the age-old question of, will the clicker actually work for me? Something happened. Okay. Yeah, okay, that's you, isn't it?
Oh, wait, no, it's going.
Okay. I don't know, I don't want it to keep going.
No,
See, this is, yeah.
all right.
PhD folks, thank you very much. For those of you who are not intimately familiar with the world of aquatic invertebrates, what are they anyway? Well, basically, the aquatic part is pretty easy. They live in the water. The invertebrates part is they wear their skeleton on the outsides of their bodies, and they do not have a backbone. So this is going to be some creatures that are probably fairly familiar to you. A lot of those fall under the heading of insects, but there are a lot of other little, often disregarded creatures like worms and leeches and mites and snails and freshwater mussels, which are abundant in the Johnson Creek Watershed, by the way. And they live in all different kinds of water bodies. And that's one of the reasons that they're really useful when you want to have a look at, okay, we did this restoration project. What did it actually do for the creatures that live there? The thing that makes them very convenient objects of study is that you can find them not just in every different type of water body, from really, really wonderful and pristine to the mankiest ditch that you could imagine, though still be something that's an invertebrate living in there, but you also find them in every different kind of part of the habitat. So if you're looking at the shoreline, there's a this cool little dude right here. I don't know if this is, people ever seen anything like that? There's a big-eyed toad bug. So oftentimes it's very easy to mistake them for toads because they're kind of bulgy-eyed and they hop around a lot, but they like to live at the margins of the stream. So if you're walking along like the edge of the like the sandy river, just kind of look down and see if you see these little dudes jumping. They can live on the surface of the water like water striders. They can live up and down in the water column. They live on plants and wood and rocks and substrate and interstitial spaces, so you find them absolutely everywhere. And you can find them far away from the water as well, because for the insects that live in the water, most of the adult versions have wings. Doesn't necessarily mean they're terrific flyers, but it does mean that you could find them in places you wouldn't necessarily expect to see them. And the thing that makes them useful to look at for restoration monitoring is that there's just huge diversity of insects and other invertebrates, right? They're really what run this planet. And there's so much diversity that they have a lot of different niches that they fill, and they have a lot of different preferences, right? So just like any animal has different conditions that it prefers to live in, it does better in, it does worse in. You get that with these different types of insects as well. So they might be associated with still waters or flowing water or thick sediments or bedrock or boulders. They might be really tolerant to pollution. They might be really intolerant. They might be sensitive to a lot of disturbance in the stream or in the riparian buffer. And so they're going to respond differently to conditions in different types of streams. And the kind of insects that we see living in these different areas kind of gives us information about what the area is like that they're inhabiting. So this entire project is done as part of the Clackamas River Focused Investment Partnership. For the purposes of brevity, I will be calling this the FIP from here on in. And it is a whole lot of different organizations and agencies that work in the Clackamas and Johnson Creek basins in those watersheds. And there are a lot of different restoration projects that have been implemented, are being planned, were implemented this year, will be implemented. And this whole span of the project covers about 6 years. And obviously the goal is to restore and enhance riverine habitats. And with most of the projects that are done, this is of course for the benefit of salmonids, right? To increase the areas where they can spawn, to increase the rearing habitat, the refugia that they can find. So it's all about fish, right? So what does this have to do with aquatic invertebrates? Everything. The answer is everything. Because everybody eats them. right? So they are at the heart of every food web, whether it's terrestrial, whether it's aquatic. You can see in this little wheel of photos here that you see aquatic organisms, terrestrial organisms, songbirds, waterbirds, shorebirds, reptiles, mammals, everything eats them. And of course, fish eat them as well. So you can make an absolutely beautiful restored reach of stream. You can look at that habitat. You can look at that riparian zone. You can say, wow, this would be a great place to be a fish. But if there aren't any invertebrates there, they don't have anything to eat, right? So it's very important to know, are there a lot of invertebrates in the system? And they do a lot of other great things in general for streams. Again, that every organism that lives in the stream or the wetland or the spring, it's not just streams, although that's what's being restored here, they're great at breaking things down. So there are some types of riffle beetles, for example, that feed only on wood and they help break wood down. Or they might eat plants or dead bodies or other sorts of things that fall into the water. They affect how stable the substrate is. So these little western pearl shells, one of our types of native mussels that can occur very abundantly in the Johnson Creek Watershed, they bury themselves in the substrate. And so if there's a flood that comes down, if there's a high flow time, if it's springtime, they can actually help keep the boulders, the cobble, the sediment in place a little bit better because they are part of that substrate. They're helping to stabilize it. They move up and down, these insects do. They burrow, they crawl, they dig, they go out, they come back in. And so they can help mix up sediments. And that's good because it helps with nutrient exchange between the sediment and the water. And it also helps get oxygen into those sediments as well. And there's a lot of critters that live in those sediments and they need oxygen and they need food. So really, really important in nutrient mobilization. And they can tell us a story because they're always there. And when they occur, they can be really, really, really abundant. So it's not hard to find them. It's not really a needle in a haystack kind of thing. They are fairly straightforward to collect and ID. And in the situation for this particular project, as far as I'm concerned, they are really, really straightforward to collect because I'm not the one doing it. Noah is. So through the blackberry. Yup, but it's worth it. So you can see this person here is holding what we call a D net or a D frame net or a kick net because the metal opening that holds the net looks like a letter D. You disturb the material in the substrate, current sweeps it in. Sometimes you collect it into 14 jars, you know, sometimes you don't. And the identification, although it's obviously it takes expert knowledge, there are a lot of different resources out there for the taxonomy. So you can figure out what these things are. And they're always in the habitat. So that means that, yeah, maybe the Johnson Creek Watershed staff gets a call. There's something oily that I'm seeing floating on the surface of the stream. Oh, I'm looking over a bridge. It looks like maybe somebody dumped something in the stream. And they can come out and they can take some water samples. And maybe whatever was being dumped is still there. And maybe they know what it is to try to analyze. But you know, there's a lot of other types of non-point source, you know, things, not as evident as something purple dripping from a pipe into the water, right? And so if you don't take a water sample at that particular moment in time, you're not going to be able to find what the stuff was. But these creatures live their entire lives in the water. They're always integrating all these different stressors. And that might be natural changes in the environment, whether it's a drought or a heat dome or a wildfire, or it might be all those different human-related impacts that we put into things, right? So if you take a look at who's living here, what are the different members of the community, right? Kind of like, you know, think of all of us here as actually being like, you know, black flies that have hunkered down on a rock or something, right? That would tell me something about what that reach of the stream was like if we were all black flies and we were all just sitting on the cobble. So changes in who's present, who's absent in the community from year to year can tell us a good story about what's happening in the habitat. And that's the hope is for these restoration projects that are going on with the FIP, that we can take a look at changes in the macroinvertebrate community pre and post restoration to get an idea of what's happening ecologically, not just physically. You can take pictures, you can count the number of surviving live stakes that you put in, but how are the critters actually responding? Because that's the whole reason for doing the project, right? Not just to make it look good, but to restore those ecological functions. And the insects are a big part of those ecological functions. So in stream restoration, there's this idea that it's actually officially referred to as the field of dreams hypothesis. That if you build it, they will come. Of course, if you make this very, very pretty 100, 500, 1,000 foot stretch of stream, well, of course the fish are going to come back. Of course the bugs are going to come back. Won't they? Too long didn't read, it doesn't always work out that way. Because you have to think about I always have to hedge this around. When I talk with clients, I'm like, no, I can do this work for you, but you have to understand there might be this to think about, and there might be this to think about, and there might be this to think about, because if you've got, let's say, a really degraded, manky, trashed up, terrible stream reach that doesn't have very much in the way of bugs living in it, and you think, I'm going to restore this, and it's going to look great, it's going to be full of stoneflies. Well, it's not going to be full of stoneflies if the nearest stonefly is 10 miles away. Right? So you've got to think about, what are my potential colonists and where are they going to come from and how far away are they coming from? And what does it look like in between where they are and where this nice new beautiful piece of habitat is? Because not all winged insects are created equal. Flame skimmer, this is one of the types of dragonflies that the dragonfly monitoring program this year and in other years has gotten to see. Dragonflies are really strong flyers. We've got migratory species of dragonflies that make monarch butterflies look like, little weenies. They actually fly a lot further. Nobody likes to talk about it, but it's true. And then I like to talk about it, but then I got to think about monarch butterflies. And then you've got these giant stoneflies. And if you've ever seen these in the flesh, in the chitinous flesh, they're like an inch or more long. And you'll think, wow, that's an amazing stonefly. But stoneflies, they got those big wings, but they take off and they kind of feel like, man, I'm a little bit faint. I need to sit down again and take a rest. And for things like caddisflies, if there's a bridge in the way or a culvert, especially if the culvert is full of spiders and spider webs, they don't disperse very well. So just because something has wings doesn't mean that it's going to be able to move really well and really far. So there's different barriers to movement for these critters. How stable is the habitat? One of the hallmarks, especially if you're turning a stream that was pushed into a ditch into a nice re-meandered sinuous kind of stream, is that the habitat changes. It's really dynamic, and so it's going to be really changeable for the creatures that live there. And if you've got just a 100-foot reach of stream that you've restored beautifully, and it's in the middle of a housing development, warehouses, old glass factories, whatever the case may be, well, the good work that you did at that small scale might be overwhelmed by all those continued stressors and insults that are still entering in. This is probably sounding like a real downer, Daniel, for this year's report, but just because you improved the stream reach, it doesn't mean that the stuff that was kind of more tolerant of heat or sediment or pollution or whatever, they're not just going to look around and go, wow, this is way too nice for me now, I think I better leave. And so you've got established creatures that, incoming colonists that might be more sensitive, the kind of critters you quote unquote want to see, they might not be able to establish a toehold very well. So there's a lot of different factors at play. And usually a degraded habitat, it happened over a long period of time. And to expect that overnight, you're going to get this beautiful ecological recovery just is not reasonable. This is actually one of our dragonfly monitoring sites. It's got greater, import than that, but this is Mitchell Creek at the Centennial School District property. And so you can see where the stream was kind of put into a new bed, and here it is, not very vegetated, but we got these nice boulders, we got some flow, now it's getting vegetated in. So this is year 1, year 2, and year 3, and it looks very different again this year. It kind of makes sense that who is living in the stream between year 1 and year 3 and year 5 is going to change, right? It's not going to be the same. And those changes in what the community looks like can be really informative. So this slide is probably going to be totally illegible to you, but what I want you to do is to take a look at the extent of the orange dots. These are all the different monitoring sites. This is what poor Noah had to do season after season. And so you can see that the first ones here are Austin Hot Springs, and then coming all the way up into like Estacada, on into like Milwaukee, and then deep into the heart of Southeast Portland. And you might also be able to see that a lot of these little orange dots are sort of occurring in pairs. And so what was done here is what is basically known as a before-after control impact study. which means, first of all, that we're doing our best to get 1 or more years of data at these restoration sites before the project goes in the ground. And then also monitor for multiple years afterwards, which is the absolute best way to get biologically relevant data. So collecting and analyzing this data multiple years pre and post project, collected in mid to late summer when the stream flow is low. And then each project, as far as possible, has been paired with a control. And talking about reference sites and control sites is a whole different matter, but we tried to find streams that were similar in setting, in topography, that we knew or hoped would not be experiencing any restoration related changes. So in other words, we can look at the changes that happen pre and post restoration in this particular stream, and then what's happening at a really similar stream nearby where restoration isn't done. Because if you see big changes in the restored stream and you see the same big changes in the other stream, you're like, well, maybe that's just climate or something. But big changes in the restored stream, and that's not happening in the control, that's a response, more likely to be a response to the restoration project. And then, because we always want to know that NOAA is actually not goofing off and doing his job well, duplicate samples were taken at 10% of the sites for quality control. And this is actually a kind of a rule of the Department of Environmental Quality, that just to kind of see how little or how much variation there is. And the duplicate samples that NOAA takes are always the most similar to each other. So again, good job. So this is just to kind of show you what NOAA is out there doing. We've got 100-foot reaches of stream, and they're divided up into these evenly spaced transects. And then he goes out there with that Z net, and at one spot, 1 foot squared space in each one of those transects, moving from left, center, right, center, left, center, right. So kind of moving along the different parts of the stream, kicks up the substrate, records what kind of flow is it? Is it a riffle, a run, a glide, a pool? What's the substrate like? Is it sediment? Is it sand? Is it cobble? Is it bedrock? Is it plants? Is it wood? How wide is the stream from wetted edge to wetted edge? And what's the depth, right, where that net went down? So you get a little bit of an idea of what the setting is like. And then all 8 of these different little individual nets are composited into one bucket, one sample. That's the snapshot of life in that particular bit of stream. So what do I take a look at then when I get the data and I'm taking a look at what all the different critters are that live in these different places? Well, obviously I'm looking at the community composition. So how similar or different? That's how similar or different is this one particular site from year to year to year to year, or pre versus post restoration, or how similar or different is the project and the control site, or all the project sites versus all the control sites. A lot of different ways to look at it. And I look at how many different species there are. What's the relative abundance? You know, you could have 2 different samples and they both have 10 species in them. But if 98% of all the different individuals in sample number 1 is all black flies, that's telling a very different story than if in the other sample that has 10 different species, only 14% of all the individuals are black flies. I know that there's a lot more disturbance happening in the first sample, for example, that maybe it's very, very recently been restored, because black flies are very often brand new colonizers. And so you'll see a lot more of them in year 1, and then those numbers just go down. So relative abundance of the individuals of the different species. How many non-insect species are there? Because generally speaking, if you have a lot of worms, if you have a lot of fingernail clams, if you have a lot of flatworms, it's not really as healthy or as intact of a habitat. So if there's a whole lot more non-insect types than insect types, probably still kind of a degraded habitat. And then of course, the big 3, the EPT, that is Ephemeroptera, Plecoptera, Trichoptera, or mayfly, caddisfly, stonefly. Always kind of considered the gold standard of stream restoration, because they like water to be well oxygenated, fast moving, and colder. In other words, they like riffles. A little bit of a complication though. We're in the Willamette lowlands. How many of us work in streams that are just lousy with riffles? So if you've done macroinvertebrate monitoring and you're like, wait a minute, what's this evenly spaced transect stuff? That's not, no, you're supposed to sample riffles, aren't you? No, not necessarily, because if you think about the kind of work that's being done to improve habitat for salmonids In some cases, it's been rehabilitating side channels or creating new side channels or putting in large wood structures to create pools that give deep, cold places for the fish. Well, that's slow water. You wouldn't expect riffles, right? And if you've got a whole reach of a really lowland stream and it has maybe one riffle and you're like, well, that's a riffle, I better take all my sample there. That's not really a representative picture of who's living in that entire reach, right? So that's why we were doing multiple different habitats, multiple different flows, because we wanted to get a much better idea, not just of, oh, do we have a riffle and what's living in it, but who all is living there. But still do take a look at those more sensitive EPT. And then I also take a look at the ecological traits, because this is what's going to change if there's more sediment or less sediment, faster flows, slower flows, more plant material, less plant material. I take a look at things like how they eat, what sorts of flows do they like, what kind of temperature ranges do they thrive in, what are the gill structures like? You know, if you've got something with really feathery, delicate gills, those can get smothered or damaged by too much fine sediment. So those organisms are going to leave. They won't be able to survive. How do they move around? If you do have a lot of fine sediment, for example, if you make your living by burrowing down in the substrate, you're going to love it if it's really, really sedimented. You're not going to love it so much if flow restoration has cleared away sediment and now you're looking at these nice clean cobbles. You're not going to be able to hang out there. So even how they move can be kind of revealing. And how quick do they go from egg to adult? Because if you've got nothing but creatures they go through 2 or more, or 3 or 4 generations per season in a site, that kind of tells you this is maybe a really disturbed site. They need to be able to get out of Dodge quick. The faster they can go turn into adults, the faster they can move on. Conversely, if you have something that takes 2 or 3 years to get from egg to adult, and you're finding those, you're like, this is a more stable habitat. It's perennial, it doesn't dry down, it doesn't have a lot of disturbance. And so this is just sort of a summary saying, well, different stressors can impact different traits. So you sort of see this here. We're doing the biological monitoring because all the different stressors have different impacts. They sort of filter the bugs, the critters a little differently. And there's going to be some response. And tolerant organisms will respond differently from sensitive organisms. And so these are just a few more examples, you know, organisms that shred material that falls into the stream. Well, if you don't have very intact riparian buffer, if you don't have you know, if you've got green lawn or something down to the stream, there's not really much there for them to shred. So you're not going to see a lot of shredders. I already talked about if you have more disturbance, you're going to get more mobile, faster developing organisms. So these are the sorts of things that, you know, haunt my nights is how are these different communities? So what does the data look like so far? This year's data is not back yet. So this is from the first 2 years of monitoring. So remember, this is a 6-year project total. Thus far, in those 2 years, 183 unique taxa. Now, that might be the level of genus or species. Some groups are left at the order of family because they're just too much of a mess to identify further than that. Most of them have been insects, also some non-insect types like these worms and side swimmers. The number of different taxa per site, the richness you can see is really, really, really different. So some of these really heavily impaired urban streams, they've got maybe a dozen different organisms, different species. You go down to Austin Hot Springs, you got 60. So very, very different conditions. The greatest diversity, the most kind of speciose groups are actually the caddisflies, as well as the true flies. And a lot of times people don't get super excited about seeing non-biting midges. People are like, oh, midges, they're really tolerant. But actually some of them are and some of them aren't. And so that's where a lot of the diversity comes from, is these little non-biting midges. But a lot of these are very urban streams, and so it's not a big surprise. There's a lot of common tolerant taxa that we're finding. Stoneflies, which are arguably the most sensitive of all of these aquatic organisms, were absent from 20 of the, I think it's 42 sites NOAA now that have been sampled, something like that. Yeah, just some total. Yeah, so 40 some sites. 3 sites had no mayflies or stoneflies or caddisflies, so again, much more degraded. Lots of collectors, so they'll filter little particulate bits out of the water. Lots of scrapers racing their way along the biofilm on the rocks. Not very many shredders, might be telling us a little something about what it looks like at the margins of the stream. If you look at all the control versus all the project sites, though, the controls have more richness, more diversity, more EPT, and fewer non-insect taxa. So that's kind of telling us like, oh yeah, this is good. The control sites actually are in better shape than the project sites. Weirdly, they also have significantly higher community temperature optima, which is may or may not be significant. Again, this is from just a couple of years of data. And at this point, we only have pre and post project data from 3 different projects, right? But they were kind of different types of projects. And thus far, again, giving you the warning, it's only 1 year pre and 1 year post. So this is still a very short time frame. The outcomes have been a little bit different. So one of the first ones that was done was on a private property just off of Foster Road on Kelly Creek. And it was the removal of a small dam. The water had really backed up behind it, right? So data was collected before that dam was removed and after it was removed. And you can see what some of the differences are. So there were 0 riffles pre-project, and there was 1 after the dam was removed. I mean, that's not huge, but it's a riffle. You know, it shows a habitat change. All of the substrate was just sand and silt. Wow, some cobble got revealed after that dam was taken out. Almost double the number of organisms, telling you that the habitat is now more heterogeneous. There's more different spots for critters to come into. There were 0 of those mayflies, caddisflies, stoneflies. 3 isn't very many, and there were no stoneflies, but that's still a big difference. It was mostly worms before, not very surprising. And now you can see that that most abundant organism is not quite so overwhelming in the entire community, and it's non-biting midges, which, you know, they can still be pretty tolerant, but it's better than worms. Lots more tolerant and sediment tolerant organisms pre-dam versus dam removal, and the number of burrowers really went down. So even though the overall taxonomy, the overall communities were still about 2/3 similar, you can see that there are some differences in the traits that are telling us that habitat really did change. Now, in contrast, we had some large wood that was put in on Lower Johnson Creek. Now, remember, that will be that sort of habitat, create refugia and pool type of habitat for the fish. And not very much of a difference in the number of taxa, pretty much identical EPT, pretty much identical relative abundance of the top taxon, although again, it was non-biting midges after the project instead of worms. A few sensitive, sediment sensitive taxa showed up. So that's kind of an improvement. And the number of tolerant organisms fell a lot, and so did the number of sediment tolerant. So again, just looking at the taxonomy doesn't necessarily tell you the whole story, because you say, wow, 66% community similarity, that's not a huge difference. And maybe there wasn't that much change in some of these different organisms. Dear God. But there were some changes that are telling you that the sediment situation is changing, the particulate matter is changing, maybe the disturbance level of the site is changing. So still some differences there. And then the last project that's gone into the ground and been monitored pre and post implementation was some engineered riffles that were put into North Fork Deep Creek. It's kind of behind a gas station in Escateda, called Casadero Natural Area. And my understanding from NOAA is that the actual engineered riffle is really only a small part of the entire reach that's being sampled, so not like necessarily a huge impact. So again, when this riffle went in, not a big difference with the EPT, a little increase in richness. Boy, still really the home of the worms. But the overall community temperature optima, so this is all the different temperature associations of all the different groups in it. It's a weighted mean, is a little bit lower, and that's encouraging because bringing the temperatures down is one of the goals of these projects. And the community sediment optima, the fine sediment optima went down. That means that the total community is not quite as tolerant to high levels of fine sediment. And so again, an improvement there potentially in some of the sediment conditions. And again, not very many other differences, and still that same community similarity, but very different outcomes.
Can you tell me what a riffle is?
Oh, God bless, yes. So a riffle is where the water is flowing very quickly. It's churning a little bit. It's a little foamy. It's breaking, usually over cobbles or rocks. And then a run is where the water is still moving pretty well, but the surface of it is much less broken. A glide is moving water, but it's kind of moving slowly. And then a pool is just a pool, very little flow. Okay. That's a good question. So this year was year 3 of data collection. There'll be 3 more years after this. I'm going to continue examining annual community variability and putting that in context with what are we seeing because there was a wildfire or a heat dome or an incredibly cold, wet spring versus what are we seeing because of the restoration projects that went in. And that's always one of the challenges. And always, again, you know, I always have to kind of hedge this by saying we have to take a look at the setting of these streams because some of them are extremely, extremely urbanized streams with impacts that aren't going to go away. There are housing developments, there are light industry, there's, you know, what have you. And so in some cases, there may not be a huge change because those landscape level stressors might end up being more important. So that's the sciencey part of the evening. I hope that it was at least to some degree illuminating. And I know there's more raffle prizes to come, but if you have burning questions, I'm going to be around, so you can always hunt me down.